Before we start the next session, which will be moderated by Buddy as well as Abby on CRISPR, which I know everyone's excited for, I figured I'd do some polling because I knew there'd be butts in the seats after Buddy's speech and CRISPR topic. So for everybody who has your mobile devices in our family, we're going to do just a few short polling questions before we start this session. Okay. I think we can pull up the first polling question. And this first one is actually for those who have been diagnosed within the last 3 years. Did your physician discuss the benefits and risks of starting corticosteroids, including potential benefits of early treatment, at your first clinic visit following the diagnosis of Duchenne? Give everyone a minute. Okay. I think we can actually give it one more second and move on to the next question, which will be the last question before we start. This is a word cloud. We know the care breakouts are coming up, and we want to know, looks like it's not responding yet, but what is one area of care you hope to get the most insight about during the care breakouts? This is for everybody, parent, person with Duchenne or Becker, family member. We know we have those breakouts later on today, so we're interested in what area of care you really hope to walk away with the most information about. Give everyone a chance to. Stretching and PT, supplements. A lot of different areas of care people are hoping to walk away with, which is good. This also gives those who are presenting on these topics a heads up on some of the things you should probably focus on. So I'll send you a screenshot of this to the docs later on. You know, it's going to be helpful. Good. Keep going. Okay, I know we're running a little bit behind, so Abby's just going to kick us off for this session. Buddy's going to be back up here as well to help with the moderation, and we can start the topic of CRISPR, which I know everyone's very interested in.
Good morning, everybody, and welcome to our last day of the conference. And I think, I wish Buddy were here because I think he had some comments to make about CRISPR, but in the interest of time, since we're running a little bit late, I'm going to, CRISPR has been part of our gene therapy initiative. It's a little bit farther out than some of the other AAV-mediated deliveries that we heard about in the past couple of days, but we are very anxious and looking very excited about these forward-thinking therapies and strategies. So we have two speakers today. We have Dr. Melissa Spencer from UCLA, and she's going to give us a little bit of an overview about CRISPR and then talk a little bit about her work. And then we have Dr. Charles Gersbach from Duke University, who will speak also about CRISPR and what he's doing and his path to bringing something forward and getting into the clinic. So I'm going to introduce Dr. Spencer now.
Hello. So I'll be talking about CRISPR today. How do I change the slides? So CRISPR is a method to edit DNA in live cells. And there have been a variety of approaches developed over the last 20 years to accomplish this goal. But CRISPR, being the newest, has really revolutionized the field because of its ease of use and its relatively increased efficiency and its wide-ranging applications. All right, so CRISPR is composed of two components that I will refer to as guides or flags and scissors or Cas9. So the way it works is you design guides or flags that can go to any region of the DNA. They can mark it, and they guide these scissors over to that site to make a cut. So it cuts both strands of the DNA. So anywhere you put a flag, you get a cut. So how it would work for DMD is if you imagine the DMD gene like puzzle pieces that have to fit together in a certain way to make productive dystrophin. In DMD, the DMD gene is missing a puzzle piece, and so it no longer produces functional dystrophin. And the way CRISPR works is you can put your flags around that puzzle piece that's missing and remove just the right number of puzzle pieces to allow those end pieces to now fit together and then make functional dystrophin. And you have an internally deleted, smaller dystrophin, but it's functional. And so CRISPR can be used similarly to exon skipping, and it can remove a single exon by placing flags around it, and there's other ways that this can be accomplished that Dr. Gersbach will talk about. But CRISPR is also able to delete any size or region of the DNA that, that one would want, depending on how these guides are designed. So why would you want to delete more than you need to? If you can just skip one exon or delete one exon, why would you want to delete a larger amount of the gene? Well, it turns out that we have learned from Becker muscular dystrophy, in which patients have these large deletions, large in-frame deletions, that the type of dystrophin that's produced can be more or less functional depending on how these endpoints come together and which part of the gene is deleted. And it turns out that one of the more functional dystrophins and one of the milder phenotypes of Becker is a 45 to 55 11 exon deletion. So what I'm showing you here is the normal DMD gene. I'm sure you've seen this many times today through the past couple of days. The DMD gene, which has a stop mutation, or in this case, doesn't produce functional dystrophin, and then a Becker muscular dystrophy mutation missing some central region of the molecule. And it depends on those balls, which are spectrin repeats. It depends on how they're framed that would partially contribute to the functionality of the dystrophin. So taking out more in some cases is actually better than taking out less. And so it just turns out that this 45 to 55 region of the gene is also a hotspot of mutation. So you can see this graph where we're plotting frequency against against each exon, and you can see that there's a peak. So not only is this region a very good region to delete to produce a very functional dystrophin, but it's also where about 50% of the mutations reside. And so with this idea, we set about developing a platform that could create this mutation using CRISPR. So Courtney Young joined my lab in 2014, and we partnered with my colleague April Pyle and set about seeing how we could use CRISPR to make this very mild BMD mutation applicable to about 50% of patients. So one of the reasons this was attractive to us is because, as you've probably seen if you've been in this field with exon skipping or any of the new drugs, each new drug needs to go through the clinical trial pipeline. So what we were hoping to do was to develop a single pair of guides that would be able to be developed that would sweep through 50% of the mutations. So we validated this platform in human cells and showed we can recreate dystrophin using two flags that flank this region, the 45 to 55 region. And you can see from this Western blot that when we reframe the DMD gene, we produce a dystrophin that's truncated about 66 kilodaltons. So that's a very large deletion. It's probably one of the largest deletions that's been made with CRISPR, certainly on the DMD gene. And we were concerned that perhaps this was not a good goal based on the level of efficiency we were achieving. we weren't sure how we compared to others. So we wanted to test this, and we received some funding from PPMD, and this is work that's ongoing, so this is a preliminary result. And I should mention that Hope for Gus Foundation helped to support this work through PPMD. So what we did was we designed a bunch of different guide pairs flanking similar exons that made BMD mutations. Or no? Uh-oh, what happened? Did it not work? What's funny? Oh, like what did I do? So you can see across the top, there's 3 different Becker mutations. There's the one we're trying to make, and then 2 others. And we made guides that were closer to the internal exons and further away. So you can see, starting from the right side of the graph, the 708 kb, that's our deletion, and we're looking at the level of efficiency. And then right next to it is a different pair of guides that are 344 kb, and there's not much of a difference there. Now, if you move all the way to the left, you can see that if you delete 1 kilobase, so that's 1,000 puzzle pieces, it is more efficient than the next most efficient, which is 100 times larger. So there is some loss of efficiency as you go down in size, but at a certain point, it doesn't matter all that much. And so we use these data to encourage us that our guides were reasonable and that it was well worth it to be able to encompass 50% of patient mutations with a single pair of guides and make this very functional dystrophin. So now we're very interested in translating this platform, and we've been doing this now for a while, and we're taking 2 approaches. The first is ex vivo editing. So you take patient cells, and they could be patient-derived, or they could be human-induced pluripotent stem cells, and you edit them in a dish, and then you transplant them. And this is primarily being done by my colleague April Pyle. Or we're also attempting both non-viral and viral delivery methods. The non-viral methods are being done in collaboration with a nanotechnologist named Wan Meng at UCLA. And so we're trying both of these approaches, but we're much more interested in trying to develop the nanoparticles for a variety of reasons. I think you heard a lot yesterday about the immune response to AAV. So for microdystrophin gene therapy, that's an issue. It's going to be even a bigger issue for CRISPR-Cas9. And I think the number I have is a little higher than what Dr. Byrne had yesterday, but it's estimated 50, 60, I have up to 70% of people have pre-existing immunity. So for older patients, it's going to be very important to deal with that. And it's also very important for CRISPR to be able to redose. It's less important, I shouldn't say that. It's more important to be able to redose for CRISPR than it is for AAV microdystrophin because of the level of efficiency that's being achieved. Also, the 2 main types of scissors or nucleases that are being used right now with CRISPR, there's also existing immunity to these components. So CRISPR is bacterially derived, and therefore it's sure to induce an immune response. So you're adding an additional, what we call immunogen, additional component that's going to trigger the immune system. So it's especially important that we consider this fact when dealing with CRISPR and AAV. And finally, AAV can integrate into the cut site. This has been shown by multiple labs, including Dr. Gersbach, who's going to be speaking next. And so we don't know what the consequence of that integration will be. Most likely, it's going to mean that the type of mutation that is created, or the type of correction that's created, cannot be very, very precise. And what I mean by that is that if one were trying to edit in a specific region, and one needed to accomplish that editing in a very, very precise way, then having AAV integrate is going to disrupt that. Now, most of us have made our guides in an area called introns, which is non-coding, so that might be less of an issue. But it is something to consider. So what are the benefits of non-viral delivery, such as nanoparticles? So first of all, they can be they can be modified to increase their functionality, and there's many ways that this can be accomplished, and I'll talk about some of them in my talk. They're biodegradable, and more importantly, they can be re-administered. So with an inefficient system like CRISPR, that's going to be very useful. But one of the challenges we've faced is how do you deliver such a large plasmid or mRNA? So nanoparticles have been used in humans. They're approved medicines, but not many have been able to deliver such a large plasmid as we need with CRISPR. So nanoparticles can be made from a variety of materials, organic or inorganic. And there's now at least 50 approved nanomedicines, probably mainly for cancer. So we've been developing a polymer-based system called polyrotaxene, or PRX. And this system is made up of a PEG backbone. So this is polyethylene glycol, and it's a very useful nanomaterial that has a lot of high stability when being delivered through the blood. And we thread onto this polyrotaxane these sugar molecules that are positively charged. And in the case of our design, we've added an extra arm of polyethylene glycol to improve the solubility, not the solubility, the stability in the blood. So when the nanocarrier is added to the DNA, I should mention this, some of the ways we can modify these nanoparticles is we can change the number of rings, we can change the number of charges on those rings, so the relative amount of binding of DNA can occur that way. We can change the amount of PEG, so in our case we've added this extra arm, and we can functionalize with peptides or linkers and change the ratios of plasmid to nanoparticle. And so what you can see here is when we add the plasmid to the nanoparticle, they self-assemble in the tube and they make this structure that looks like that ball at the bottom. So I'm just going to briefly talk about some of the changes we've made to improve our nanoparticles. One is the addition of a disulfide linker. So what we found early on is our plasmid wasn't being released when it got into the cell. So by adding this linker, it helps the nanoparticles sort of fall apart. We've also added peptides, and we've tried to target stem cells. Targeting muscle stem cells will greatly improve this therapy because it would make it much more long-lasting, because these cells can actually divide and pass the edited DNA onto a daughter cell, where that does not occur in differentiated muscle. And we've shown that we can put these nanoparticles in with our plasmid and obtain editing. And there's multiple steps that are involved. Once that nanoparticle enters the cell, they have to traffic, they have to come into the cell, they have to release from what's called lysosomes, the plasmid has to go to the nucleus, and then it has to be expressed. So we're always checking in vivo how the intracellular trafficking, in vitro, so in cells, we're always testing that intracellular trafficking. But then when we ideally design our nanoparticles, we then want to go in vivo and see how they perform. And what I'm showing you is just a variety of different nanoparticles that we've been trying, and I'm showing you a bunch of different muscles, and that got shifted a little on the top. And what we found, if we label our nanoparticle, and it looks red here, we can see if they're going to muscle after being delivered throughout the body. And we had good uptake, not as great as we'd like, but we did have good uptake of several of our nanoparticles, except one that performed really well in vitro didn't perform so well in vivo. So this adds another challenge, is if you make a modification that helps one aspect of your trafficking, you might hurt another. So we're testing these now. We're at this point are obtaining very little dystrophin, and so this has a lot of development still to do. But it's very exciting for us, and we think it's worth the long-term investment to get these nanoparticles working. But in the meantime, we're wondering, how does our platform perform using AAV, which is what most CRISPR scientists are using right now in their therapy development. So other groups have shown that they can use CRISPR to deliver, they can use AAV to deliver CRISPR in a mouse model of DMD. And so we partnered with Jeff Chamberlain, who generated some AAV for us with our platform, and we used a muscle-specific CK promoter, and we ran our guides in a separate, we have to use 2 AAVs because the entire system doesn't fit into one. So that's another issue that we are facing, is that by having to use 2 AAVs, we effectively have our dose. So we also used mCherry so that we could track it, and we found that if we used AAV6, delivered it systemically, we could see dystrophin produced, and it was of the right size. I'm not going to show the Westerns right now. And we use a separate antibody to dystrophin to eliminate revertant fibers. So anything that's within our cut region would eliminate a revertant, and then we would know that that was not developed from our platform. And we generate about, in this case example, so these were mice injected at 5 weeks of age, about 10% wild-type dystrophin, about 31% dystrophin-positive fibers from a single injection. We think that we would like to do much better, and we would like to be able to redose. This is not a level we would ever take into clinical trials. And we've also used AAV9 as well, which is the popular AAV right now. And this also creates, we didn't count yet how many positive fibers, but we quantified the dystrophin, and it's actually less, it's cut off a little bit. We're getting about 5 to 6% dystrophin. And of course, you cannot compare these two at all. I'm just showing 2 examples because they were done, these were done with a different dose and a younger mice. So I'm just showing that proof of principle, you can make dystrophin based on our platform. It's still pretty low, and we're working to improve that and to be able to redose. So our platform targets 50% of patients. We're using viral and non-viral delivery methods. We can tell that based on our preliminary results that CRISPR is much more efficient not much more efficient, it's more efficient over very short distances, but as you move out from a very short distance, the efficiency doesn't change tremendously. And we'd like to continue to develop our platform, which is a very large deletion, because we think that it's beneficial to be able to target more patients and to just use redosing to increase the efficiency or the efficacy. So we are also working on ways to get rid of Cas9, because this is going to certainly trigger a myositis or some sort of muscle inflammation if it sticks around, which is what the AAV episome does. And we need to, very nicely establish off-target effects, and we need to do functional testing, which is what we're doing now. And based on PPMD funding, we're doing the Cas9 killing and the high-fidelity nucleases, so high-fidelity Cas9. So Dr. Barry Byrne gave me this slide to use because I wanted to sort of put in perspective where we are. So he mentioned yesterday that I'm sort of hovering those those listings above because we don't know exactly where we are. We won't know till we get to the end, but Dr. Byrne pointed out that AAV microdystrophin is sort of where the rain is right now in that steep uphill climb. And I think CRISPR is far behind right now. We don't know, maybe we'll catch up, maybe we will go at the same pace of AAV microdystrophin, which took, a good 15 years, 20 years to develop. So I'd like to just acknowledge this is a multi-lab collaboration, and some of the collaborators are shown on the top. The work on the nanoparticles was done by Michael Imami and Courtney Young, but Courtney Young started this project as a PhD student. I'd like to acknowledge my colleagues at UCLA, which are a little bit cut off, and Jeff Chamberlain. Thank you. And my funding, our funding.
Okay, thank you to the organizers for the opportunity to come and tell you about some of the work that we've been doing at Duke University in developing gene editing for DMD. And thank you to everyone in the audience for taking the time this morning to come out and hear about this. This is a disclosure slide I'm required to show by my university, and the main point here is that this, all the work that I'm going to show is part of a really fantastic partnership that we've had with Sarepta Therapeutics that has been supporting all of the work that I'm going to show and really allowed it to move forward at a level that otherwise wouldn't be possible. So I think I correctly predicted that Dr. Spencer would give a fantastic introduction to gene editing and into CRISPR, so I'll probably skip over a lot of the details to talk more about some of the nuances that we're trying to address with our approach, but I think as she already introduced, the whole idea behind gene editing is to correct the mutations that occur in the genome or correct some form of, restore some form of a functional dystrophin gene in the genome rather than providing the gene on an AAV vector, and I'll talk along the way about some of the differences between that. And I don't think I need to introduce Duchenne or the types of dystrophin mutations to this audience, but I'll just give a little bit about my thoughts about why we decided that Duchenne was going to be a really interesting and important target for gene editing technologies. And of course, that starts with the clinical need that needs to be addressed. But I think there's some other technical considerations. So as opposed to some of the mini or microdystrophin projects, the ability to correct the form of the gene that already exists in the genome could potentially provide a means to give more of the protein back to the cells and to the tissues, a larger fragment of the protein. The potential to edit genes in satellite cells. So this was referred to a couple times yesterday, and I'm going to talk more about this a little bit later, but the idea that you could correct the stem cells that occur in the mature muscle tissues could provide a continuous source of new dystrophin protein expression that would lead to long-term dystrophin restoration. Muscle tissue is a really interesting target for gene editing, both the cardiomyocytes, because there's a potential for a kind of one-and-done therapy, since those cardiomyocytes basically exist for the lifetime of the patient, but also because the skeletal muscle fibers have hundreds of nuclei within each cell. And so, as Dr. Spencer mentioned, a lot of the gene editing approaches right now, particularly for in vivo gene editing, have relatively low efficiencies, but the ability to correct only a few of the nuclei, so a few copies of the genome within each muscle fiber, but then have that nuclei produce dystrophin to compensate for all of its unedited neighbors creates a real opportunity to restore dystrophin expression in skeletal muscle tissue. As Dr. Spencer mentioned, the ability to remove exons and restore a functional protein is actually an easier strategy for gene editing than to deliver exons back, although I'm going to present some data here in a second about some of the strategies that we're taking to restore full-length dystrophin. And then also, as was mentioned earlier, the ability to address large segments of the gene so that you can develop a single CRISPR composition or single gene editing approach that can get to a large segment of the patient population is very exciting. So there's a number of different ways to edit these genes. Fundamentally, gene editing is about creating a break in the DNA and then taking advantage of DNA repair pathways to create different types of sequence changes. The first one on the left is using a method of DNA repair that's called homology-directed repair. And the idea there is that you break the DNA and then you provide a template that has some sequence similarity to the target site in the genome, and therefore some piece of DNA can then get copied into the DNA break, and that piece is shown in purple there, and that allows you to insert things into the genome. However, that pathway is relatively inefficient compared to other pathways, particularly in cells that are no longer dividing, like the heart muscle cells and skeletal muscle cells. And for that reason, most of the efforts that are being explored use these two strategies on the right hand of the slide, either making a single break in the DNA And then after that single break is created, random nucleotides of DNA can be added or removed from that target site that would allow you to either disrupt certain sequences, like sequences that are involved in including exons in a mature dystrophin mRNA, or shift to the reading frame. So if you add or remove a base pair or two from the DNA, then an incorrect reading frame could be shifted back into the correct reading frame. And then there's a strategy shown on the right that if you wanted to just completely remove an exon from the genome, you can make 2 cuts on either side of that exon, which would be basically reproducing exon skipping at the genome level. And I'll come back to that in a minute. So there's many ways in which you can create these targeted breaks in the genome, as Dr. Spencer mentioned, but the kind of most exciting and most recent is CRISPR because it's such an easy and robust technology to use. And because so many people are using it, it's also led to many different types of adaptations and improvements in using this to treat disease. The CRISPR-Cas9 system has 2 components to it. One is the Cas9 enzyme. So this is a protein that's shown as the big blue blob in this cartoon, and then the guide RNA. And so the guide RNA is a short nucleic acid that's shown curled up in the pocket of the Cas9 protein. And the guide RNA is what takes Cas9 to go and find its target site in the genome. And once it finds a similar sequence to what's present there in the guide RNA, it docks onto the genomic DNA where Cas9 is then activated to do this cutting that then stimulates DNA repair. And so I'll focus first here on this idea of creating one break in the genome, or what has sometimes been referred to as kind of a single cut gene editing approach, and the idea that you could add or remove a few base pairs of DNA at a single site and then use that to either disrupt splicing sequences or reframe a disrupted gene. And so we explored this concept many years ago, first with an earlier technology of gene editing called TALENs, and then later with CRISPR. And we started with patient cells that were correctable by exon 51 skipping, but rather than removing exon 51, we created these breaks within the exon 51 sequence that would allow us to add or remove a couple of base pairs of DNA and potentially reframe the expression of a functional dystrophin protein. And so this is the Western blot shown on the bottom left, shows how we were able to restore dystrophin protein with this approach. And then on the right shows the sequences that were generated that added or removed different base pairs and then enabled reframing of the correct gene. This is with that earlier TALEN technology. We later did the same thing with CRISPR. And again, you'll see the list of different sequences provided on the right, some of which were able to restore dystrophin protein expression. So this is one of the earliest things that we did, and I think it's nice because of the simplicity of creating a single cut. There were a number of concerns that we had with this type of approach, and one of those is that, as shown on the right, the types of sequences that come back come from random insertion or deletion, or I'd say semi-random insertion or deletion of base pairs. And the fact that some of those would create reframing events and some of them would not, and then even those that would create the reframing events would create random new amino acids in the protein. And so we were a little concerned we could never really predict exactly what the function or immunogenicity of all of those different epitopes would be. And that because it's random, every single cell would get different assortment of those epitopes. We decided to explore something that would be would generate a more consistent protein from the same editing event. And that was the idea that if we could remove a single exon, that we should always get the same protein back rather than a random assortment of new epitopes. And so in this strategy, we went back to those same cells, and then we created 2 breaks, one on either side of the exon that should always remove exon 51. And there were a number of different reasons, some of which Dr. Spencer referred to as to why this kind of double cut on either side of the exon and removing it we thought would be advantageous. And one, as I already mentioned, this NHEJ DNA repair mechanism that removes segments of DNA from the genome is relatively more efficient than homology-directed repair that inserts things back into the genome. So removing things or deleting things is always going to be more efficient. I already referred to the fact that by making a double cut in the non-coding regions around the exon, the regions that don't encode for the dystrophin protein, we didn't have to worry about creating unpredictable protein products. Also, if we're targeting within the exons or at the exon splice sites, you're really limited to just a few, maybe 1 or 2 or 3 guide RNA options that you can design in that sequencing range. But in the intronic regions around the exons, there's hundreds of thousands of base pairs. That gives you great flexibility in what guide RNA, what targeting molecules you might use. And we really wanted to take advantage of that targeting space to enable us to create the most efficient, but more importantly, the safest and most specific guide RNAs. And so we've developed strategies for screening thousands of guide RNAs in that sequence space to come up with the very best gene editing reagents. And then finally, as Dr. Spencer already mentioned, that going beyond just a single exon, where if you're trying to reframe or disrupt an exon, you need a new CRISPR reagent for every single exon that you want to target. In this case, we can do this large exon 45 through 55 deletion with just one Cas9 and two guide RNAs with a single CRISPR composition that could address about half of the patient population. So I'm not going to go over too much some of the work that we did exactly in that area. Much of this has been published, and much of it is very similar to what Dr. Spencer already described. This is just to summarize some of that. We started with patient-derived cells. We showed we could use these exon deletion approaches, both for a single exon or the larger 45 through 55 deletion that would be applicable to half of all of the patients. We did that first in cell culture. Then we did some work in the MDX mouse model where we corrected the expression of the mouse dystrophin protein by removing mouse exon 23. And with Dongcheng Duan at University of Missouri showed that we could restore muscle function or improve muscle function in that mouse model. But then to make CRISPR reagents that can target the human gene and not just the mouse gene, we went out of our way to create a humanized mouse model of the disease that carries the full-length human gene with specific mutations that we can introduce with CRISPR and then come back and correct with therapeutic CRISPR reagents. And so that work, some of you have probably seen us present before or has already been published. So I wanted to talk about 4 key points that I think are going to be important for either addressing some of the questions in the audience or advancing CRISPR forward towards a clinical candidate. And those are the issues of immunogenicity, as was already mentioned, durability, which is related to immunogenicity, and as related to durability, the ability to target satellite cells that would lead to long-term dystrophin restoration, and finally, the restoration of a full-length dystrophin product. So first, around immunogenicity, we use a similar approach as was described earlier of using AAV to deliver the CRISPR reagents to restore dystrophin protein and improve muscle function. So that work has been widely described. More recently, we wanted to look back and better understand this issue of immunogenicity of the bacterial Cas9 protein that's been widely discussed. And so we first look at an antibody response to Cas9, and the way that we designed this experiment was we either treated these adult mice with the AAV CRISPR system or neonatal mice with the same system. And what we observed was that we see a very clear, robust antibody response to Cas9 when we treat the adults. We do not see that same antibody response when we treat the same mice 2 days after birth. We also wanted to look at T-cell responses, and since we're delivering the Cas9 with AAV and not directly into the blood, we actually aren't that worried about an antibody response. What would be more concerning is a T-cell response where these immune cells might attack the cells that are expressing Cas9. And in fact, again, when we treat as adults, we do see T cells that are specific for Cas9 that were induced by this strategy. And again, we don't see that same response when we treat as neonates. And so the idea here is that the underdeveloped neonatal immune system does not mount this response against the bacterial protein. And while we don't know that this would actually be reproducible in a human setting, we think that it does point to some ideas around immunosuppression that we might be able to use to avoid this type of immune response. And there's other things that could be done to the vector to restrict Cas9 expression that may also help overcome this issue. Now, of course, an immune response to what we're delivering as the therapy, this CRISPR-Cas9 system, is not desirable. We were also given the number of different studies that have come out showing positive results with CRISPR gene editing, understanding what is the impact of this exact immune response on the actual therapy. And so we wanted to better understand durability, and is this immune response affecting durability? Again, we did either an intramuscular injection to adults or a systemic administration to neonates, and then we took data at either 8 weeks post-treatment or at 6 months or 1 year for the neonates. And while the intramuscular administration to the adults, we did see a decrease in gene editing between 8 weeks and 6 months. With the systemic administration, we actually saw no decrease in editing over time and actually saw increases in some tissues. So this suggests that there may be ways to circumvent the immune response, and even with the intramuscular injection, we still saw relatively high levels of dystrophin expression all the way out to 6 months or 1 year. And this is showing the level of DNA editing, but this histology shows the levels of dystrophin that we were seeing out to 1 year and even increased over what we were seeing at 8 weeks. So even though we are seeing an immune response to Cas9 that you would expect to a bacterial protein, we don't actually fully understand what the implications are going to be for affecting the treatment and to what extent that immune response might be able to be controlled by immunosuppression, similar to what's being done for other AAV-type gene therapies. So we were very excited to see that we were getting durable restoration of dystrophin all the way out to 1 year in these treated mice, but it also begged the question about why is that happening? Why are we seeing such long levels, knowing that these muscle tissues are regenerating and that this regeneration happens through satellite cell division and then integration into the damaged muscle fibers. And so probably many of the audience are familiar with this, but satellite cells are the stem cells of skeletal muscle. They exist all the way through adulthood, and they sit on the outside of the muscle fibers. And in response to exercise or muscle damage, those satellite cells will divide and then fuse into the muscle tissue. And that is how muscle regenerates and repairs, and it's also how muscle gets stronger by adding these new nuclei. Now the concern there, and this was discussed a bit yesterday, is that if the nuclei or if the genomes within the muscle fibers are turning over and being replaced by satellite cells, then even if you edit the genomes in the muscle fibers, then you may not get long-term durable dystrophin restoration if they're being replaced, unless you're also editing the satellite cells. And so we wanted to ask the question, does AAV get to satellite cells? Can you use AAV CRISPR to edit the satellite cells in vivo, and does that lead to long-term dystrophin restoration? And if you were listening to some of the conversation yesterday in the gene therapy session, there's been actually quite a bit of controversy about some of these questions for several years now, where there's a lot of speculation as to whether AAV may or may not be getting into satellite cells. And one of the problems is that once a cell divides, it will lose the AAV vector. And so that's fine in things like skeletal myocytes, cardiomyocytes, and other cells that are not dividing, and that's why you get long-term expression of microdystrophin or other genes from those AAV vectors. But if the AAV did hit the satellite cell, and then the satellite cell divided, as it would in a regenerating tissue, then it would quickly lose the AAV vector. And so if you simply deliver AAV and then come back a couple weeks later and look to see if the satellite cell is carrying AAV, it might not be there, even if it was there previously. So to better understand this in a really precise way, we developed a system in which the genomes of the satellite cells would be marked permanently if they had ever been exposed to AAV. And so the experiment that we did used these mice in which all of the satellite cells are labeled with a green fluorescent protein. So if we look at their fluorescence, we can see they're green. And this piece of data here shows that when we sort out all of the single cells from the muscle tissue, about 5% of them are GFP positive, are green, show green fluorescence. And that's about right with about the frequency of satellite cells that you would expect in skeletal muscle. So then the question is, when we deliver the CRISPR system to the skeletal muscle, and then we purify out these green satellite cells, are they in fact edited? And in this case, we did this in the context of this MDX mouse where we would be removing exon 23. And we were very excited to see, this is showing 3 different mice looking at the GFP negative cells or the GFP positive cells. And in every case, we went and removed exon 23 at some frequency in the satellite cells, and we can sequence it and see it's exactly what we want. So that was really reaffirming to us, and this reproduces a lot of pioneering earlier work by Amy Wagers at Harvard that had originally showed some of this as well. So now across multiple labs, and now Dongsheng Duan has shown similar results, we're seeing editing of these satellite cells in vivo. Now that was a very new observation, and I think addressed some of the controversy in the field about whether AAV was getting to satellite cells by using a method that was easier to track them. But interestingly, it got us thinking about which AAV serotypes might be useful for getting into satellite cells, because there's many different AAV serotypes available, and the ones that are being used right now have been selected based on their ability to get efficiently to muscle fibers. But no one is really picking AAV serotypes based on their ability to get to satellite cells. And so we went back and revisited the idea of which AAV serotypes might be best for getting to satellite cells. And to do this, we used a different system that was going to be easier to work with than even CRISPR, in which we start with the mouse that all of the satellite cells are labeled green. And then we deliver an AAV that carries an enzyme that if it goes into the satellite cells, it's going to turn them red. So the question is, after we inject the AAV into the mouse and collect up the satellite cells that are green, how many of them have turned red? Those red cells would be, that are dual positive for red and green, would be markers of satellite cells in which AAV vectors had got into them. And in doing this, we did this first with a local intramuscular injection of the AAV vector across 6 different serotypes that you see here. And we were, again, really excited to see that 60% of the satellite cells were turning red following administration of some of the commonly used AAV serotypes, including 9, 6, and 8. And so this was very exciting to us. It also addressed some of the questions that have come up, and this was also discussed yesterday, of, sure, maybe some of the satellite cells are being edited, but how do we know it's not some rare population of proliferating satellite cells or satellite cells that aren't regenerative and already might be on their way into the muscle tissue? And that was actually something that we thought originally when we saw some of the early data as well, but showing that 60% of the satellite cells in a single muscle are being edited by this AAV vector, I think is really promising that we're getting to a large number of them and that this could be a promising path forward. And that was with a local administration. When we did systemic administration and took out 5 different muscle tissues, we were seeing editing rates in the satellite cells between 20 and 30%. Again, a number that we were really excited about and pleased with. We can also take these tissues, we can go in and take sections of them and do staining and see, okay, are these edited satellite cells that are expressing these green and red proteins where they belong in the muscle tissue, hanging out outside of the muscle fibers? And the answer is yes, they are. And then finally, a key question is, if these are really, truly regenerative satellite cells, then you should be able to take those edited satellite cells out of one mouse, put them into a damaged muscle in another mouse, and have them contribute to muscle regeneration. Because we really wanted to prove that these are satellite cells that are self-renewing and continuously participating in muscle regeneration, which is not an easy experiment to do, but in this case, we collected up those satellite cells after we had treated the mouse with CRISPR, and then moved them to another mouse that never saw AAV or CRISPR, and showed that in fact those cells could contribute to dystrophin restoration and dystrophin-positive fibers, and we could also do the molecular analysis to show that those cells that are missing exon 23 are present there. So we do think that these are not just differentiating cells, but are actually cells that can contribute to regeneration and potentially even long-term self-renewal. Okay, last question that was, I think, mentioned yesterday is, what is the potential for restoration of a full-length dystrophin protein? We know that the microdystrophins produce a truncated version, and as has been presented today, everything that you've seen has been using gene editing not to give back a full-length dystrophin gene, but to actually remove exons, one or more exons, and give back restore a partially truncated, partially functional protein. So I mentioned that the strategy on the left for adding segments back to the genome is very inefficient, and that's partly because it uses particular molecular machinery that recognizes the sequence similarities between the gray piece and the green piece, the gray piece being the one that you deliver to get into cells. And that's inefficient compared to the strategies on the right, where really all the cell has to do is grab the 2 free ends and ligate it back together again. That's always going to be easier for the cell to do. So we wanted to develop strategies in which we can maybe fool the cell into grabbing a different set of free ends that would go into the genome, free ends that we're going to generate from the AAV vector, rather than using this other pathway that's boxed there on the left-hand side of the screen. And so this is building on other results that use what's called homology-independent targeted integration. And so to give you an idea of what that looks like, The top piece there shows what a wild-type gene would look like between exons 50 and 53. Now, I mentioned earlier that we created a humanized mouse model that carries a full-length human gene, but is missing exon 52. So we created a Duchenne-like genotype by intentionally removing exon 52. So now exon 51 and exon 53 don't fit together, and you have a disrupted reading frame. Now, the strategy that we designed was to make a guide RNA that would target that sequence between 51 and 53, where 52 is supposed to be. And we made two AAV vectors. One of them encodes the Cas9. The other one has that guide RNA that targets the sequence between 51 and 53. But also on that AAV vector is exon 52. And exon 52 is also flanked by that same guide RNA sequence. So if everything goes as planned, we deliver these two AAV vectors to the cells, that Cas9 plus guide RNA is going to go cut that target site in the genome between 51 and 53. It's also going to cut the two sites on either side of 52 and release it from the AAV vector. And then if we can fool the cell and provide enough of this vector, and it grabs those two free ends on either side of exon 52, rather than grabbing the free ends but on either side of 51 and 53, then we can ligate that 52 back into the genome where it belongs and restore a full length of dystrophin gene. So we did this, we delivered those AAV vectors to this humanized mouse model of Duchenne muscular dystrophy, and then we can use PCR to explore, did 52 go back into the genome where we would expect it to? And in fact, it did. And you can see we tried it with a number of different vector doses and ratios and guide RNAs, and in all cases, we get different levels of this missing exon going back to its home in the dystrophin gene. And that led to restoration of full-length dystrophin protein by Western blot in these muscle tissues. Now, this is still very early work, and we have a lot to do, and we still have to understand the efficiency of this approach and do some optimization, but I know there's a lot of interest in this community about what potential approaches might be available in the future to restore a full-length dystrophin gene, and we think that this might be one of them. So just to summarize, these genome editing strategies for DMD typically focus on removing gene segments to restore a functional truncated dystrophin. We and Dr. Spencer and others have developed approaches where with a single gene editing composition or a single approach, it would be applicable to restoring functional dystrophin in about 50% of patients. We do observe anti-Cas9 immune responses, as has been widely discussed since it's a bacterial protein, but we also see long-term dystrophin expression, and we've never seen any adverse consequences of these immune responses in any mouse model that we've treated. And in fact, many of the immune responses seem to kind of self-resolve over time without any intervention or immunosuppression. Of course, there's going to have to be a lot of work in large animal studies to understand what better understand what that might look like in humans. I didn't have time to talk about some of the different methods that we've developed to interrogate other types of gene editing events that might be happening. That's important, but we think that it could possibly be controlled. And then we're really excited about this idea of new strategies that could restore a full-length dystrophin gene, but it's going to require some more work. And in particular, across all of these CRISPR approaches, there really needs to be considerable more investigation to immune response, the effect of long-term presence of the delivery vector, and the alternative genome modifications. But nonetheless, we're working very hard and very excited about it. I try to highlight each of the students and postdocs in my lab along the way that worked on the different projects, but we're greatly appreciative of all of the different foundations and agencies that have supported our work along the way, and in particular, this very, very fruitful and productive partnership with Sarepta Therapeutics that has supported this most recently. Thank you.
Thank you both. That's really exciting work. I think that Buddy's talk this morning, for those of you who are here, was really appropriate because he talked about having to go through the service room or take the back stairs, but there is always a way to get there, and hopefully we're going to find a way to get there too. We're in that middle stage, finding all the different ways. We have time for a very few questions right now, so come up to the mics, and I think that Buddy has the first question for Dr. Spencer and Dr. Gersbach.
Yeah. Now, Melissa and Charles, I hear both of you use the term efficiency a number of times. And I was just curious, how exactly do we define efficiency in terms of, is it the repeatability of the technique, the delivery, ultimately the effectiveness? Just a clarification, I guess.
Yeah, go ahead.
Yeah, it's an important and somewhat complicated topic. I mean, I think that even efficiency of dystrophin expression just in general has its own nuances, and which methods that you're using and how you're quantifying them, and that only gets more complicated when you add a gene editing component. When we think about efficiency, I think there's issues that there's multiple levels of this. There's how efficiently are you delivering the system to the cells? How efficiently is it modifying or editing the DNA once it gets to the cells? And then how efficiently do those edits of the cells lead to dystrophin production? And all of those things require different methods or have different methods that could be used for each of those. And all of the numbers are a little different. So to give an example, in our mouse studies, we had kind of looked pretty carefully with different methods at each of those different steps. And what we have seen is that we are delivering the machinery in mouse models to almost all of the cells, but we only saw editing at the DNA level in the range of 2 to 3%. However, because as I mentioned, because it's a multinucleated tissue and there's hundreds of nuclei within each fiber, 2 to 3% editing at the DNA level led to 70% of the muscle fibers expressing some dystrophin protein. And I say some dystrophin protein because 70% tells you how many of the fibers, but it doesn't tell you how much dystrophin is being made by each of those fibers. When we analyzed that by Western blot, the answer was about 8%. So it's complicated, but got 2% efficiency of editing at the DNA level, led to 70% of the cells expressing dystrophin, and the average across all of those 70% of those fibers was about 8% of normal dystrophin levels. That was in our earliest preliminary mouse study, and obviously we're improving upon those numbers now.
Yes, sir. So my name's Kyle Lico. My son is 3 years old, and he has a duplication of exons 10 and 11. And I just was going to see if that's something that you thought could be possible in the future in regards with gene editing, and if that would be, I know that adding back in sounds like maybe less efficient than just deleting. Is that something that you see possibility in the future to fix?
It's definitely technically conceivable, right? So, I mean, the idea, just as we showed removing of exon 51 or exons 45 through 55, a similar approach could be used to remove exons 10 and 11, I think is what you said was duplicated. There is an extra challenge there, which is removing one copy of 10 and 11 while leaving the other copy behind. Because the sequences are similar, there's the possibility that you would remove both copies, and I don't have the puzzle diagram memorized well enough to know what happens when you remove both. But so that does add a challenge for duplications, but I think that there's other strategies that you could even consider as well. For example, adding 1 through 9 at the front part. So there's a lot of clever things that people are coming up with. So I think that it's possible in the future that any number of different possible mutations could be corrected by this approach.
Thank you.
And I'll just add that while CRISPR has a lot of versatility, there are some regions of the gene we wouldn't want to do without. So the work of Jeff Chamberlain showed, for example, the region that hooks dystrophin up to the membrane. It's a very small region, but if you cut that out, you still have a dystrophy. And so there is a lot of applicability, versatility, but there's also, there are some limitations still.
Gotcha. Okay, thank you.
I may have a similar question, but I want to ask it anyway. My nephew has a deletion 8-9. So would that be the same answer that you just gave to this gentleman for 10-11?
He's a deletion of 8 and 9?
Yes.
So, I mean, the strategy that I showed, for example, of adding 52 back could be applied to adding 8 and 9 back. But I guess I want to make clear that there needs to be some work done in understanding what the pathway is going to be. If and when gene editing becomes mainstream for treating lots of genetic diseases, how unique gene editing reagents will be developed for different patient-specific mutations is, I think, something that is a new challenge from a regulatory perspective that is going to need some need some work to figure out what the best path forward is for that. But certainly, it's conceivable to use gene editing to add those back. There's issues of efficiency, as I mentioned, but.
Okay, thank you.
Okay, so thanks again very much to our speakers this morning. I think we're going to move right on to the next panel. So if we could have the panelists come up.